Skin cancer ad campaigns have been around for decades that raise awareness regarding skin cancer. But how deadly is skin cancer? Let's find out. In this video, we are going to be looking at skin cancer. We are also going to be looking at the etiology behind the disease, epidemiology, pathophysiology underlying the etiology, and the signs and symptoms of skin cancer. Skin cancer is the uncontrolled growth of abnormal skin cells. It occurs when unrepaired DNA damage to skin caused by a variety of factors triggers mutations or genetic defects, resulting in skin cells rapidly dividing and forming malignant tumours. Melanomas are dangerous because they have the ability to metastasize. That is, they have the ability to spread to other parts of the body, such as the lungs, brain, or liver. There are three layers of the skin: the epidermis, the outermost layer, the dermis, and the hypodermis. Skin cancer is found in the epidermis. In the epidermis, there are squamous cells, melanocytes, and basal cells. These cells respectively cause their corresponding skin cancer. Basal cell carcinomas are the most common skin cancer and generally grow slowly on the head, neck, and upper torso. It may appear as a lump or dry, scaly area. Squamous cell carcinomas have the potential to metastasize and spread to other parts of the body if not treated. It grows over months and appears on the skin most often exposed to the sun. It can be thickened, red, scaly, which may bleed easily or ulcerate. Melanoma is the most deadly and can become life-threatening in as little as 6 weeks if untreated. It can appear blotchy on the skin not normally exposed to the sun, and it is usually flat with an uneven, smudgy outline and blotchy. It is also more than one colour: black, brown, blue, red, or grey. Skin cancer is a major health issue in Australia, with an estimated two-thirds of Australians being diagnosed with skin cancer by the age of 70. It is estimated that in 2016, 13,280 new cases of melanoma was diagnosed in Australia. Males represent a higher proportion of new cases than females. 24% of those with skin cancer are aged 60 to 69. For basal cell carcinomas, the age standard incidence rate was 884 cases per 100,000 people, and for squamous cell carcinomas, the age standardised incidence rate was 387 cases per 100,000 people. For Indigenous Australians, there is a lower risk of being diagnosed with skin cancer than non-Indigenous Australians, with 84 cases of melanomas being diagnosed in Indigenous Australians, and the age standardised incidence rate for melanoma was 9.3 cases per 100,000. A main cause of melanoma is excessive ultraviolet, or UV, exposure. The two types of UV radiation is UVA and UVB. Another cause of melanoma is genetics, such as having fairer skin or a weakened immune system. UVA oxidizes the melanin in the dermis, which is produced from melanocytes when exposed to UV radiation, producing a dark pigment, whereas UVB damages the skin's superficial epidermal layers and causes direct DNA damage. UVB are the main rays that cause sunburn and most skin cancers. As such, melanin is released by melanocytes and acts as a barrier and filter to UVB, reducing the penetration of UV through the epidermis and protecting the skin. Sunlight is the main source of UV rays, along with tanning beds and sun lamps. So how does UV radiation actually cause skin cancer? Well, the UVB exposure causes a pyrimidine dimer to form from thymine bases in DNA, leading to cross-linking in melanocytes. This leads to oncogenes to be activated and tumor suppressor genes to be inactivated, and inhibition of DNA repair. The most common oncogene that is activated in melanomas is the BRAF gene becoming mutated. The BRAF gene is part of a stimulatory pathway which regulates cell growth. Once the gene is mutated to BRAF V600E, the melanocyte overproduces regulatory proteins from continual activation of the stimulatory pathway, leading to increased cell proliferation, decreased cell survival, and inactivation of apoptosis. The V600E is a missense mutation that changes valine into glutamic acid and is the most common BRAF alteration and constitutes for 90% of BRAF activating mutations, or 50% of metastatic melanomas. In regards to tumor suppressor genes, the CDK2NA gene is commonly found in melanoma. This gene controls cell growth and aims to prevent rapidly uncontrolled cell division, and therefore, if it is mutated, melanocytes grow exponentially, which is highly dangerous. Another tumor suppressor gene that is alterated in melanomas is P53. The P53 protein is activated by cell stress or DNA damage and induces apoptosis. When the role of P53 is disrupted, the melanoma is not destroyed and proliferates. Once uncontrolled proliferation of the abnormal melanocytes occur, they enter the radial growth phase where they grow horizontally. They then enter the vertical growth phase and grow vertically. The melanoma then presents itself on the skin surface and may penetrate the dermis and the subcutaneous skin, known as the metastatic phase, and has the ability to access blood vessels, enter the bloodstream, and go to various parts of the body. The signs and symptoms of melanoma are best understood through the ABCD rule. A stands for asymmetrical shape, where the melanoma lesions are irregular or not symmetrical in shape. B is for border. Typically, non-cancerous moles have smooth, even borders. Melanoma lesions have irregular borders, so difficult to define. C, colour. The presence of more than one colour or uneven distribution of colour is a warning sign for melanoma. D, diameter. Melanoma lesions are often greater than 6 mm in diameter, about the size of a pencil eraser. And finally, E, evolution or change. The evolution of the mole is the most important factor to consider when it comes to diagnosing a melanoma. Knowing what is normal is life-changing.